But we're going to finish this up. We'll do most of the wet stuff from Lab 12 on Wednesday. We'll do blood typing and some other things on Wednesday. But for this evening, I want you to pull out the slide of the blood stain, and you want to be able to identify the different blood cells. We'll start first with the erythrocytes. So the red blood cells are all these pink cells. It should be easy to identify. As you scroll around, you'll see these little, and it's not really in focus, but see these little dots? These little purple dots? Those are your platelets. This doesn't have a whole lot.
The screen does.
I know, right? I thought those are the platelets.
Here's one. So there's a few on this slide, not a whole lot. This person here is sick. So this is an individual with leukemia. So the reason why I put it up here because a couple of the white blood cells are kind of stand out. The other thing you're going to be looking for are the different white blood cells on your slide. The first one, these really large ones, are your monocytes. And the key to the monocytes, identifying them, is look at the size compared to a red blood cell. They're about twice the size of a typical red blood cell, or even larger. So they really stand out as being much larger than your typical red blood cell. Also on here you have lymphocytes. Notice that the lymphocytes are about the same size as a red blood cell. So they look similar if you look at just the nucleus and the little cytoplasm, but monocytes are much larger, whereas lymphocytes are about the same size. Now it also shows on here a neutrophil. So down here we have a neutrophil. Neutrophil, eosinophil, and basophil are all what are known as polymorphonuclear cells. They have multi-lobe, multi-shaped nuclei, which is what you see here. 1, 2, 3. This one has 4 lobes on it. So one of the key things you're looking for will be these multi-lobe nucleus on each of the cell types. Then the difference between neutrophils, basophils, and eosinophils are going to be the identification of the granules inside of the cell. Neutrophils, you don't see any granules. Okay, so all you see is basically a little bit of light cytoplasm and the multinucleated or multi-lobe nucleus. That's a neutrophil. On an eosinophil, you're going to see a similar structure, but you're going to notice that there's a bunch of these red granules. A bunch of look like little red speckles or dots inside of the cytoplasm. Those would be the eosinophils. And then the basophils, it'll look similar, but the cytoplasm will be filled with all these dark little purple spots or granules. So that's how you're going to be able to differentiate between these three. Neutrophils, you're not going to see the granules. Eosinophils will have kind of red granules or kind of pinkish granules. And then the basophils will have more of a purple granules inside of them. And so that's pretty much what you're going to be looking for on these slides. You want to be able to differentiate monocytes from lymphocytes, eosinophils, neutrophils, and basophils, okay, from each other, as well as erythrocytes and then the occasional thrombocyte or the platelets. Okay, so you work on differentiating between those, and then you can also, on page 80, you can do a differential white blood cell count. Just to kind of get an idea of how many or what percentage of each of the white blood cells that you might find in blood. Okay, so you're going to go through and scroll through and start counting white blood cells and identifying which ones they are. Count 100 total white blood cells and try and determine what percentage of each that you should find within your blood. I'll give you a hint. You're not going to find hardly any basophils, if you find any. You might get one. If you're lucky, you'll have 2. Eosinophils, you'll find a few, maybe 2 or 3. The rest you should be pretty numerous, so you should be able to find quite a few of those. Okay, but eosinophils and basophils, there's not that many on the slides. They're more difficult to find because they're not that abundant in the blood tissue. Okay, so you'll only see a couple of those. But that's what I want you to work on this evening. So I want you to take care of the cats, and then I want you to look at the slide of the blood, and then finish filling out the table, review your slides from the endocrine system, and that's pretty much our focus for this evening.